Before I walk you through our programs at Precigen, I would like to mention that I will be making some forward-looking statements, and for those, I will refer you to our most recent filings for the descriptions of these statements. As you might know about Precigen, we are a cell and gene therapy company that really has unique technologies all under the same roof for not only the constructs for the expression of various number of genes, but the various platforms for delivery, non-viral and viral, and also the most important thing in a gene therapy, how do you control the expression? And we have a number of switches, and one of the most advanced switches in the clinic that allows us to basically bring all of these components together and address the various treatment for the patients. In the next slide, these are the three main platforms that currently Precigen is advancing. Our differentiated Ultracar T platform that uses a non-viral gene delivery method, and we are capable of delivering a number of genes under this one expression system, which allows us for a drug homogeneity. And also, we can now transfect these cells under 24 hours and basically at the site of the hospital and transfer back to the patients. We also have an adenovirus immunotherapy platform that uses a library of the gorilla viruses, adenoviruses, which they have a large payload capacity and allows us for repeat dosing. And they are very unique, and I will be discussing some of the clinical trials that are ongoing there. And finally, our bacterial platform, which allows us to genetically manipulate the L. lactis bacteria that have a great safety and express number of genes, especially for the autoimmunity at the mucosal sites. And we have an ongoing trial in type 1 diabetes with that. If we go to the next slide, this is the current clinical portfolio of Precigen with 6 assets directly in the clinic in various clinical phases from phase 1 to 2, and I will be highlighting some of this. In the next slide, our Ultracar Overnight platform. This is a very unique platform that is differentiated from a conventional CARs or basically off the shelves or TCRs. And why would I say that? In this platform, what we have done is have looked at the issues that is associated with, for instance, conventional CAR-T programs or platforms that currently exist, which uses a lentiviruses. It requires a centralized manufacturing. It takes 4 to 8 weeks with a very high cost and a high probability of failure. And it's basically, you can, with all of that, you can dose the patients once with a very high price tag. What we have done is we have used our ultra vectors to design a CAR-Ts that express not only the CAR of the interest, but also a membrane-bound IL-15, which allow persistent and expansion of these cells directly in patients, and introduce a kill switch for a safety that you can eliminate these cells at any given time. But more importantly, we have now really reduced the manufacturing to overnight at the site of the hospital by introduction of our ultraparator. This is a device, it's a semi-closed device that you can basically transfect between 3 to 4 billion T cells under 12 minutes, allowing you to scale up, go to higher doses with a minimum handling of these cells, and then putting the cells overnight, the next day, QCing them and returning the autologous CAR-T of the patient back to the patient under 24 hours. And this is extremely important because there is no other platform that allows this flexibility. In the next slide, you see the two Ultracar T clinical trials that are ongoing. The very first one in PGEN 3005 in ovarian cancer, as you are well aware, that currently none of these cell therapies have shown any promise for the solid tumors. We have targeted our Ultracar to the unshed portion of MARC16, have equipped it with the membrane-bound IL-15 to directly expand in the patient and persist, and contains a kill switch for safety. And what we showed in the first 6 patients in phase 1 that we reported, it was at the very low doses between 7 to 27 or 8 million cells. We have shown not only very promising expansion as well as persistence of these cells for months, but also in 50% of the patients, we were able to show a reduction in a total target tumor burden. On the other side, we have the PGEN 3006, our Ultracar, which targets the AML. These patients, as you know, they have very limited time in front of them. So none of the conventional cell therapies and manufacturing would be able to address, for instance, these disease. And currently we are running this at Moffitt Cancer Center, phase 1/1B, and we have reported on both arms, the lymphodepleted and non-lymphodepleted arm of this trial. And we have shown very promising expansion as well as persistence of these cells. And for instance, it was presented at ASH that one of the patients, we have passed 7 months and you still can see that these cells persist in this patient. Also in our lymphodepleted, at the very low dose, we have shown a CRI in the patient, and the patient was bridged for a transplant, which prior to that, they were not, would have not been able to do so. So we are very encouraged with this data, and we are rapidly moving toward finishing the dose escalations and phase 1B for the expansions. In next slide, this is the vision that we have for our Ultracar, that eventually we will have what we refer to as Ultracar Library approach, that when our patients come into the clinics, they get diagnosis, their tumor is profiled, and the CAR T of the interest, the oncologist can basically order that through the clean room of the hospital. This is basically manufactured overnight without any activation, without any expansion, and the next day it's basically returned back to the patient. And the reason that you don't need any expansion or activation outside is because these Ultracars, they have the mechanism already engineered in them to address this. And the flexibility of these platform is unlike anything else, because you simply can infuse and make more than one CAR T if it's needed for the disease that it's involved. If you want to redose the patient, you can bring back the patient and redose, because you manufacture these cells overnight. If you want to redose the patient with a different CAR, you can do that. The cost of this platform compared to the conventional CAR Ts is very different, and you can imagine why it allows you for the flexibility of redosing more than once and actually number of times or with different CARs. And that is what it makes this really what we refer to as precision medicine at its best. In the next slide, I would like to give you a little bit of highlights on our adenovirus platform. As I mentioned, this is a library of gorilla adenoviruses. Unlike Ad5s, these adenoviruses, first of all, have the high payload, but more importantly, you can repeat dose them, because in humans, there is either zero or very little seropositivity. This is off the shelf. You can express number of antigens and switches in them. And comparison to other vaccine, that they either don't allow for immunogenicity or they don't basically have the good immune responses. here you can have repeated dosing with the expansion of T cells. And even compared to TCRs that you have to go through a large manufacturing of number of epitopes before you can address the epitopes of interest, here you can design large epitopes into this and then directly allow the body to come up with the T cell repertoire that is specific for the antigen of interest. In the next slide, this is currently the 2 clinical trials that are ongoing, or PGEN 2009, that has used the adenovirus platform to target HPV 16 and 18 in HPV indications in cancer. And currently we're running the phase 1/2 trial at NCI under the CRADA. We have finished the phase 1 dose escalation of this trial, and we have shown a very good safety as well as the ability to repeat dose and increase the number of T cells upon every repeat in this, which makes this very unique compared to all the other platforms. We have finished the enrollment in phase 1. We also have started the combination therapy with the checkpoint inhibitor and the phase 2 in a neoadjuvant setting at this point. And we will be ready to show some of the interim data at our R&D in upcoming November 4th. Similarly, we have used this platform for PGEN 2012 in a recurrent respiratory papilloma patients. This is a devastating disease that the patient develop benign tumors in their trachea and larynx and sometimes in the lower respiratory parts of the lung. There is no option except multiple surgery, and some of these patients, they undergo hundreds of surgeries over their period of time and years. And currently, this is in phase 1. We have finished the dose escalation and have entered to the expansion phase of treatment for the patients. And we are very excited about the result, and we will be reporting on interim data in our R&D day as well. In the next slide, this is the highlight of our microbial platform, as I mentioned, the AG019, which addresses basically the type 1 diabetes. And we reported this year positive data for a phase 1b, 2a. And this molecule, basically, it's formulated to capsule. These basically lactase have designed to express IL-10 and proinsulin, that the function is to really increase the Treg activity, reduces the autoimmune T cells, and we were very excited and remain excited with this platform as we showed the stabilization of the C-peptide through the mechanisms that I mentioned, and we are currently in discussions to see how rapidly we can move this in the upcoming clinical trials after this positive data from a phase 2a. And with that, I would like to thank you for having this opportunity to present our story to everyone, and wish everyone a very good rest of the meetings. Thank you.